Hi everyone. Today we will be doing something that is very confusing to most of you. It's the respiratory balance sheets. We'll try to understand where during respiration we have the generation of different electron donors and ATP, and how we get to the final amount of 30 to 38 ATPs per molecule oxidation of glucose, right? So to start with, as you know, respiration is what? It is the oxidation of glucose. Cellular respiration, aerobic cellular respiration is the oxidation of glucose to give you what? To give you ATP, right? It generates ATP in your system. Now, mostly during respiration, ATP is generated by two basic processes. One, we call it the substrate level phosphorylation. So substrate level phosphorylation basically refers to the phosphorylation of the basic substrate, which is glucose, which leads to the generation of ATP, and oxidative phosphorylation where the oxygen is used to phosphorylate substrates and therefore give you ATP. Now, majority of the ATP that is generated during something which is known as the electron transport system, ETS. So to understand ETS, we will have to understand the the organelle structure of the mitochondrial matrix. So the mitochondria has a double membrane system, correct? So it has an outer membrane and an inner membrane. This is the internal matrix. This is the intermembrane space. Remember, in the internal or inner mitochondrial matrix, membrane, there is these 4 groups of protein complexes, right, which help in electron transport, and the 5th complex is basically an enzyme known as ATP synthetase or ATP synthase. Okay, so electron transport will happen through these 4 electron carriers, and you will have generation of ATP. So now, without getting it further complicated, let's start with the glucose that we internalize into our body. So every carb that we will consume will get digested, will get broken into glucose. Now this glucose undergoes a process called glycolysis in the cytoplasm. Glucose is converted through a 10-step process into pyruvate molecule, right? So glucose gets converted into glucose 6-phosphate, then to fructose 6-phosphate, fructose 1,6-bisphosphate, and then 2 molecules of glyceraldehyde 3-phosphate. Till here, it's known as the preparatory phase of glycolysis, where a total of 2 molecules of ATP get used. Okay. Now from here on, you have 2 molecules of G3P, which gives you 2 molecules of 1,3-bisphosphoglycerate, 2 molecules of 3-phosphoglycerate further. When the G3P, right, glyceraldehyde 3-phosphate gets converted into bisphosphoglycerate, it gives you 1 molecule of NADH plus H plus for every G3P. So you get a total output of 2 NADH here and 2H plus, 2 protons that is. And then when the 1,3-bisphosphoglycerate gets converted into 3-phosphoglycerate, it gives you a total of 2 molecules of ATP. Further, the 3-phosphoglycerate is converted into 2-phosphoglycerate, which is converted into 2 molecules of phosphoenol pyruvate or PEP, and then the PEP is finally converted into pyruvate. Again, in this reaction, you get a total output of 2 molecules of ATP. Now then, the net gain is 4 molecules of ATP, but 2 were already used in the preparatory phase. So in the compensatory phase, you therefore get directly 2 molecules of ATP from glycolysis, and you get 2 molecules of NADH and 2 protons from glycolysis, right? This calculation we will come to later on. Now moving on, to link reaction. What happens to the pyruvate is that the pyruvate then releases one carbon, so one molecule of CO2. So for two pyruvates that you get from one glucose, you get two carbon dioxide molecules. So this is one glucose that gives you two pyruvate. So now the reaction will be in doubles, right? So from one pyruvate you get one CO2, here you get two carbon dioxide molecules, and obviously two NADH plus 2H plus molecules, correct? This acetyl-CoA will then start a reaction which is known as the Krebs cycle inside the mitochondria. In Krebs cycle, acetyl-CoA actually reacts with oxaloacetate and gives you a citrate or citric acid, which undergoes a cyclic step of reaction. So now you have citrate which changes into cis-aconitate, cis-aconitate to isocitrate, isocitrate to alpha-ketoglutarate. When this change happens, when it changes into alpha-ketoglutarate, you get a release of one CO2 for one isocitrate and one NADH plus. So basically here again the total output is 2 CO2 for 2 acetyl-CoA, no? Because you have 2 pyruvate from here. So everything is in doubles, and 2 NADH plus 2 protons. Yeah. The alpha-ketoglutarate then gets converted into succinyl-CoA. During this reaction again you get an output of 2 CO2 and 2 NADH plus 2H plus, again because double, right? Succinyl-CoA gets converted into succinate, so it releases the coenzyme A and also releases 2 molecules of ATP for the 2 pyruvates that we have. Then the succinate gets converted into fumarate, where it releases yet another proton electron donor, which is FADH2. So you have 2 FADH2 from here, and then the fumarate to malate, malate to oxaloacetate. There you have another output of 2 NADH plus 2 H plus ions that are donated. So now therefore the total output from Krebs cycle will become 2 plus 2, 4 molecules of CO2 and 2 here. So you have the total of 6 CO2 that is expected, and NADH you have 2 plus 2, 4 plus 2, 6 total. You have FADH a total of 2, and you have direct ATP synthesis of 2 molecules, correct? Now what happens to this NADH and this FADH2? These NADH and this FADH2 donate electrons, correct? NADH donates 2 electrons to the first complex, protein complex. FADH2 donates 2 electrons to the second protein complex. What these protein complexes do, because you do not have the detail of electron transport chain, I'll not go into the detail of it, but what actually happens is that the first one gives 4 protons, the third one gives 4 protons, and the last one gives a total of 2 protons, releases 2 protons, right? So proton would be this. Now as a result, for every NADH, the 2 electrons that it donates in a chain formation gives a total of 10 protons, and for every FADH, for every 2 electron that it donates, it gives you a total of FADH2 gives you a total of 6 protons, right? So that is the proton output. Now what happens during the electron transport chain such that ATP is generated? So you have a proton gradient generating in the IMS, intermembrane space. The amount of proton, it becomes more and more and more, which results in the movement of proton through the ATP synthase, because there is more proton here and less here, right? So it's known as a chemiosmotic gradient, so osmosis like phenomenon with the help of chemicals. And now because of the movement of the proton, ATP synthase generates ATP. it has been found that for every 4 protons, 1 molecule of ATP is generated. So in that case, NADH gives you 10 protons, right? So for every molecule of NADH, 10 upon 4, that is roughly 2.5 ATP molecules can be generated. For every FADH2, 6 upon 4, that is roughly 1.5 ATPs can be generated. Now because it is quite confusing to work with these numbers, fractions, therefore we take the higher limit. So we have said that for 2.5 we'll consider it to be 3, for 1.5 we'll consider it to be 2. Therefore then, for all the NADH that is generated, you will multiply the number by 3 to get the total amount of ATP. So in glycolysis, because 2 NADH is generated, you have a total amount of 6 ATP. For link reaction, 2 NADH, so you have a total amount of 6 ATP. For citric acid cycle, you have 6 NADH, so you have 18 ATP. 2 FADH, so you have 4 ATP. If you calculate the total, it comes down to a 38 ATP molecule. Now similarly, if I were to calculate it with 2, then the numbers would vary such. So it would become 4, this would also become 4, this would become 12, and this would become 2. So if we take the smaller boundaries, so if we take 2 instead of 3 and 1 instead of 2, so if we take the lower round offs, then the number would become 2 plus 4, 6 plus 4, 10 plus 12, 22 plus 2, 24, 28 and 30, right? So the variation then becomes 30 to 38 molecules of ATP. I hope this much was clear. We will see you again in the next video. If you need a total recap of all of these 3 stages, then we can make a video on that as well. Let us know. Thank you.